Good evening. I'm Mary Close Oppenheimer, president of the Salisbury Forum. As always, we're very pleased to have you with us tonight. If you're new to the forum, we host nationally recognized speakers in free public forums. To be notified about our upcoming events, please go to our homepage and join our email list. You can also follow us on Facebook. If you or your friends miss one of our talks, videos are available on our website within a couple of days. It allows you to watch them at your leisure. There's some great talks there, so I encourage you to check them out when you have the time. You go to www.salisburyforum.org and look under videos. Since our talks are free, we're really grateful for any and all donations. I want to particularly thank our longtime supporters. We couldn't be here without you. They're easy to make by clicking on the donate button on our homepage. We'll have a Q&A period at the end of the talk, so please don't use the chat feature. Instead, click on the Q&A icon at the bottom center of your screen and type in any questions you may have. We'll also have a couple of poll questions popping up. They'll disappear once you answer them, and then we'll go on with our talk. Our next forum will be on Friday, September 17th, so I hope you'll save the date in your calendar. Our speaker will be Eric Edstrom, whose topic will be, what does patriotism mean anyway? Edstrom is an Afghan veteran, senior fellow at the Eisenhower Media Network, and author of Un-American, A Soldier's Reckoning of Our Longest War. He'll explore the contentious and double-sided topic of patriotism and whether or not America's current brand of patriotism is serving us as a nation. Now, moving on to tonight, if you weren't already familiar with the CRISPR gene editing technology, you may be now as a result of Jennifer Doudna and Emmanuelle Charpentier's being awarded the 2020 Nobel Prize in Chemistry for their breakthrough research. The technology allows precise edits of the genome. Researchers hope to alter human genes to eliminate diseases, create hardier plants, and wipe out pathogens and more. In light of this, we're very happy to welcome Catherine Freije to the Salisbury Forum to discuss the power of CRISPR-based technologies for diagnosing and treating viral infections. Catherine is an infectious disease scientist who has worked for the last 8 years studying human pathogens like hepatitis C virus, Zika virus, and most recently, SARS-CoV-2, the virus that causes COVID-19. We're all way too familiar with that virus. This was the focus of her PhD work at Harvard University in the lab of Dr. Pardis Sabeti at the Broad Institute of MIT and Harvard. Her work in Dr. Sabeti's lab sent her to Honduras to deploy her diagnostic technologies in country, and it was for this diagnostic work that she was recognized by Forbes magazine in their 30 under 30 list in healthcare. She's speaking to us from New York City, where she is currently working in the lab of Dr. Charles Rice at Rockefeller University. So with that, I'm so pleased to welcome Catherine to our talk tonight.
Thank you, Mary, for that kind introduction. Thank you to the Salisbury Forum for inviting me to speak tonight. And thank you all for tuning in this Friday evening. It's beautiful outside, so I'm glad that you decided to take some time inside to watch and hear me speak tonight. The Berkshire area is a spot that I've been so lucky to be able to hike, and so I'll have to get there in the next time I can. I hope, though, now that you're all feeling the same sense that I have, that we are close to and in some ways beyond the light of the COVID-19 pandemic, sort of dark tunnel. And as I hope to convince you tonight, there are amazing scientific technologies that have allowed us to get to this point so quickly, and hopefully will better prepare us for future pandemics that we may face. So the past 14, 15 months has really been marked by these scenes of emptiness, an empty Times Square at rush hour, empty restaurants, empty concert halls, empty classrooms in universities, and the empty auditorium like where I would typically have given the Salisbury Forum lecture tonight. And in many ways, when I talk to people about the last year and a half, there's this distinct memory of what we were doing the day or the few weeks before everything shut down. I remember just a few weeks before, I was traveling to Washington State and then New York interviewing for the position that I'm in now. But when all of that started happening the few days before, we did the almost like snowstorm apocalyptic run to the grocery store to get enough food for the month. And then I'm sure we all remember the toilet paper gate, where you could only have 1 or 2 packs at the store because they were worried they were going to run out. All of these measures were taken to prevent the spread of a respiratory pathogen, COVID-19, which we call it now, that we really didn't know much about then. There was really no clear guidance on how to treat this pathogen, and also many unknowns about how long it would stick around, and when and would we even be able to have an effective vaccine at some point. And I want to go back now that we're sort of at this, as I said, at the light or beyond the light at the end of the tunnel, which is that less than a year before COVID-19 came into our knowledge, we were actually talking a lot about outbreaks. And that's because it was the 100-year anniversary of the 1918 flu pandemic. And I show you here sort of 2 iconic shots from that time. So one is sort of the filled emergency hospital in Kansas, filled with people sick from the flu. And then the other is this plot of the mortality of the 1918 flu. And it actually kind of reminds me, and hopefully it reminds some of you, of a lot of the graphs that we have sort of been anxiously watching to see how cases are growing and falling in the US. And now we're in this like rapid downward trend because of the amazing vaccines that have been developed. And so what's interesting about sort of the timing of this is in the early time of the COVID-19 pandemic, we actually were talking a lot about whether this outbreak was very similar to the 1918 flu. Is this like something similar to the flu? Is it a little bit worse, a little bit better? And all of that sort of speculation was because we just didn't really know anything about this virus. We didn't know how many people were actually becoming infected early on. We also didn't really know that people that were asymptomatic or pre-symptomatic, so not having symptoms but getting them later, could transmit the virus. And so I want to say that the timing of this and is kind of brought to our attention those sort of the repercussions of not being prepared for pandemics. And I also want to bring some of you back to if you were at the last Salisbury Forum, which is that Stephen Johnson talked about how life expectancy was greatly blunted by this flu, with approximately 50 million deaths across the globe, and the fact that this virus affected sort of middle-aged 20 to 40-year-olds, which was quite unique for a flu pathogen, which usually affects the young and the old, which we were very lucky with COVID-19 that we were not affected by a virus that sort of targeted these very distinct age groups. And the other thing that I want to draw your attention to back on this plot is that there was questions about COVID-19 being seasonal, whether it was going to go away in the summer months. And we all know now that yes, caseloads dropped in the summer months because people spend more time outside, but it wasn't going to go away as we had hoped in the early days. And so we are very lucky that COVID-19 didn't really exemplify the same mortality as the 1918 flu, and that we didn't lose as many lives. But the 20th century should have warned us that outbreaks like this or pandemics like this were probably going to happen again, because the 20th century was speckled with early warning signs that something like this could happen. And so virus outbreaks were not really uncommon in the last 100 years between the 1918 flu and now. I start in 1947 with the Zika virus outbreak in Uganda, which is actually when Zika virus was originally discovered. But then we move to parts of the 70s with Ebola and Lassa virus outbreaks, as well as in the 80s when HIV hit the scene. And then we actually had 2 other coronavirus outbreaks in the early part of the 2000s with SARS and MERS. But we didn't actually stop there. And with this number of outbreaks, we saw the largest Ebola outbreak to date in 2013 that ravaged West Africa. And I was actually living in New York at the time when we were worried about US medical professionals that had come back from the front lines and with the risk of bringing Ebola back to the US. And we were lucky to sort of stop that in its tracks and get those medical professionals treatment. And then in 2015, at the start of my PhD, we saw a massive outbreak of Zika virus in Brazil that had ramifications for pregnant women and young infants. And that virus actually spread all the way to the US, where there was local transmission in Florida as well as Texas, the places where the mosquitoes that carry Zika virus live. And so even those two outbreaks that happened rather recently didn't really prepare us well enough for facing SARS-CoV-2, which emerged in China in 2019. And there are many reasons why we are seeing this increase in viral outbreaks in the last 100 years. I mean, one of the obvious reasons is globalization and the ease of travel. It's really easy to hop on a plane and go from one country to the other, and that just causes more mixing of populations and more ability for diseases to spread at a larger scale. But we're also seeing a lot more sort of new and emerging viruses. But there's always been a question in my mind that also comes from my training in Pardis Sabeti's lab, which is that, are we actually seeing new pathogens, or do we just have new technologies and the capacity to actually figure out what a pathogen is? If I look back at the 1918 flu, we actually didn't know at the time whether that was a bacterial pathogen or a viral pathogen. And it took 15 years to figure that out. And it also then took many more years to actually sequence that entire virus and know what was the sort of cause of that outbreak. And so, as my mentor always says, is it actually an emerging viral disease, or is it just an emerging diagnosis that we're able to more easily sort of catch these pathogens as they initially break out? And then the last point I want to highlight is that climate change is actually also a major player in this increase in outbreaks, in that we're living in new areas, we're forced to grow food and get food from different areas, and also viruses like Zika and Lyme, which are transmitted by insects. Because of warmer climate, those insect ranges are growing and causing sort of more areas of the US and Europe to actually have those insects that could then transmit disease. And so despite all of these reasons that seem obvious that they could increase our risk of exposure to pathogens, we were still very far from being prepared for something like COVID-19. And so I'd like to bring your attention to this quote from Ed Yong, who's a science writer for The Atlantic. And actually, around the time of the 100-year anniversary of the 1918 flu, he wrote this article that was called, "Is America Ready for a Global Pandemic?" And this article reiterated what seems so obvious and what I mentioned in the last slide, which is that we had the largest Ebola outbreak in 2015, and a mosquito transmitted virus like Zika virus could go all the way from Brazil to Central America and the US. And so he says, the epidemics of the early 21st century revealed a world unprepared. And even as the risks and the obvious events continued to multiply, much worse is coming. And what I love about this article is that he really hit the nail on the head of what could go wrong and why we were so unprepared at the start of COVID-19. Hospitals operate at close to capacity, making influx of patients challenging to accommodate. Our reserves of personal protective equipment, or PPE, are not deep enough. And we all saw the major rush for surgical masks and gloves, and the CDC telling us not to go get N95s because we needed to save them for the medical professionals. We just didn't have enough supplies. And on top of that, supplies are medical supplies are often not actually made in the US and rely on supply chains from China and India. So when those get cut off because China is the epicenter of the outbreak, then we have to rush and frantically try to figure out how to make those supplies to supply our own country. And so I will stop this sort of like Debbie Downer and say that many experts from different fields are needed to coordinate a better preparedness plan. But myself as an infectious disease scientist and not a public health expert and not a policy expert and not an engineer, I think specifically about, what are the translational technologies that will better prepare us for stopping outbreaks in their tracks? And one of the things that has and will continue to plug some of those holes that I can sort of contribute to based on my expertise is the field of genomics. And so genomics encompasses a lot of things. It's considered an interdisciplinary field of biology, and it centers around the study of DNA, which is our genetic makeup. So every single one of our cells has that. But it also encompasses the study of RNA, which we're probably now all familiar with as we've heard of mRNA vaccines and RNA viruses. But RNA is also very special in that it's the message that that tells our cells what proteins to make and has us carry out the essential functions of life. And so genomics involves the study of both DNA and RNA, and that code is made up of a very simple 4 letters, which are shown on this slide here: A's, T's, and C's and G's. And over time, as our cells grow and multiply and divide, that genetic code can accumulate mutations, which are shown randomly in red. And some of those mutations can be helpful, as we've heard with some of the new viral variants, that there are mutations that cause the virus to be more transmissible. But at the same time, mutations can also be detrimental. And so we don't see those because they get sort of wiped out by what we call natural selection. And in the last 30 or so years, it's actually become much, much easier and cheaper to get all of this information, whether it's our own DNA so that we can figure out if we're at risk for Alzheimer's or at risk for developing breast cancer. We also use genomics in the cancer field to sequence cancer genome, the genome of the cancer cells, to figure out if we can better hone treatment and what drugs may or may not work. But also we can also use DNA sequencing to figure out, what breed our mixed breed pet is. So we've really sort of opened the door for all these things that we can learn about our DNA just because of the ease and cheapness of generating this data. And I want to say that genomics has also been extremely important for infectious disease because it's important for identifying what is making people sick for viruses that we've never seen before, like COVID-19. And as I mentioned, genomics is also why we're getting all this information about the different variants of COVID. So whether it was originally the B117 variant, but now we've made it easier and named them by Greek letters. And so we've been seeing a lot of things in the news about the new Delta variant. And this is actually achieved by sequencing viruses that are in our nasal samples that we give to a lab because we want to contribute to science. And we get all of this sequencing information from it, and then we compare it to what we've seen before. And these mutations allow us, if we see them many times, say, oh, this mutation is becoming more common, and so this is a variant, and we should learn more about it and what the risks are. And I want to go to a specific example, which is that this method of sequencing viruses was very important back in 2013 during the Ebola outbreak. And that's because we know that Ebola typically introduces itself into a population by either interacting or eating wildlife in the forest, and that's called eating bushmeat. And so early in the Ebola epidemic, the messaging that was given to communities is to stay away from bats and other wildlife and don't eat them for a shortened amount of time because that's going to prevent the spread of Ebola virus. However, work by Dr. Sebeni's lab, which basically was sequencing a lot of viral genomes from patients that were getting sick in West Africa, told a much different story. The story that that the sequencing told us is actually that transmission was happening from person to person. And why is that, and how do we know that? And that's because Ebola survives in wildlife, bats and monkeys, for long periods of time. And they don't really get sick, and they don't really spread between them. And so what that does is the virus that comes from that animal looks very distinct from the virus from another animal. And so it's almost like a fingerprint. And so none of those are exactly the same. However, when a virus spreads within a population, like what we've seen from COVID-19, it accumulates mutations, but not enough to actually look that different between individuals. And so that difference between having sort of distinct fingerprints versus things that are very similar is what told researchers and told the Sabeti lab that actually this virus was spreading from person to person. And so that allowed the messaging to change during the Ebola outbreak to say, actually, we shouldn't be worried so much about bats and wildlife. We should actually be having better hygiene practices and washing our hands and staying away from infected individual. And so this is just one example of how viral genomics have really allowed changes in policy and allowed an actual change to prevent transmission. But I also want to highlight that genomics has been really important, not just for the variants, understanding the variants in COVID-19, but also has been very important for vaccines. So as you may or may not know, it was in December of 2019 when we first got word that there was this outbreak of some sort of unknown respiratory illness. And then in January, just a short couple weeks later, the full sequence of that pathogen was generated. And this is when vaccine researchers got to work and said, we're going to figure out how to get a vaccine of this as soon as we can, just by knowing the sequence alone. And that's where we get mRNA vaccines. And I just want to briefly mention that there's been a lot of talk about how mRNA vaccines are a new technology, and that we've never used this before, but that's actually not the case. That in actually 1990 is when the idea of mRNA vaccines was initially conceived. And in 2005, we were actually using mRNA vaccines to treat cancer by programming or by making an RNA that senses a cancer antigen. But then actually 2012 was the first time that we actually used mRNA vaccines for infectious diseases like flu and another respiratory pathogen known as RSV. So that's 9 years ago now. So this mRNA vaccine technology has been in the works for a long time. And that's actually what allowed things to move as quickly as they did, because we had an established technology to do this. And so in April, the mRNA vaccines entered phase 1 trials. And then in August, the phase 2 started, or phase 2/3 started. And then in December, we had the emergency approval from the FDA. And this is actually a really amazing technology, which is why I want to talk about it just a little bit more, which is that basically all that vaccine researchers do is take that sequence that they got back in January. They found the spike protein sequence, which is the protein that speckles the outside of the coronavirus. And that's the part that our immune system sees when we become infected. And they basically take the sequence that programs our cells to make the spike. They surround it with fat molecules, which we've been using this as a strategy for delivering drugs for a long time now. And that fat-coated or lipid-coated mRNA gets put in our vaccine vials. We get our shot, and then that RNA then goes into our cells and tells our cells, I got to make this spike protein. And that spike protein then gets put on the surface of our cells in our body, and then our antibodies see it and are now primed for the next time that we see the spike protein, whether it's coming from our second mRNA shot or from the coronavirus itself, and sort of helps us fight off the next time. But today I want to talk to you about some of the genomic technologies that are near and dear to my heart. And so as Mary said in the introduction, October 2020, there was also another major event that was important in the field of genomics. And so I'm going to pull up the first poll here. And it's a very simple question. It's, have you heard of CRISPR? And I will caveat this with, you can't say that you've heard of CRISPR because of my talk title or because of Mary's kind introduction. Before tonight, had you heard of CRISPR? Did you know what it was? Maybe you've never heard of it and sounds like foreign language and it's a no, or maybe it rings a bell. And so I encourage you to put in your answers.
Right now we have 88% of the people who've said yes, they've heard of it, and 3%, oh, I'm sorry, 3 people, 7% said no, and 5% have said rings a bell. So, so that means that, you know, most people are quite familiar with it.
Great. And I think this is kind of a testament to the fact that, one, we have a really a very educated and knowledgeable audience, but also the fact that this year has meant that a lot of sort of terms that scientists and biologists and virologists use are actually becoming more mainstream. I feel like Dr. Fauci has told us so much about antibodies and serology tests and interferon and things that I couldn't have ever imagined would be in the news. So I'm glad that most of you have heard of it and before tonight. So as I said, and as Mary said in the introduction, in October 2020, the Nobel Prizes were announced. And one of the Nobel Prizes, the Nobel Prize in Chemistry, went to these two amazing women scientists, Dr. Emmanuelle Carpentier and Dr. Jennifer Doudna. And give me 1 second. From this, actually, this discovery is actually another testament to the field of genomics. But before I talk about that, I want to talk a little bit about sort of why this discovery led to the awarding of a Nobel Prize and why this is such a powerful technology that has a lot of implications for human health. So as many of you probably know as well, is that CRISPR has been sort of synonymized with gene editing or genome engineering. And this is an amazing technology that would enable our scientists to be able to correct either disease states in cells or genetic mutations that are detrimental and turn them into something that is sort of the healthy state. And some of the more advanced sides of gene editing technologies to date. More in the last year or so, we've actually been able to start clinical trials for the use of CRISPR for gene editing of sickle cell anemia, as well as a congenital form of childhood blindness. And so what I want to say here too about gene editing is that we're starting to enter the era of being able to start these small scale clinical trials. And that's for specific diseases that don't have any other treatment options. And as a result, there's really just, this is the best thing that we can try to see if we can help and make our people with these diseases healthier. And so we're not at the stage where we're, using it instead of drugs that have some effect. It's more like this is a last resort. And this is because we're still trying to evaluate the safety of using this technology in humans. But I also want to say, and Mary mentioned this too, which is that we don't actually just have the potential to edit humans. We can also use this technology to edit plants, to make them more drought or heat tolerant. And the other thing that I think is a large consideration for this technology down the road is just making sure that if we're going to edit these plants or humans, that there's no sort of unexpected consequences down the line. And we saw some of the backlash of that a few years ago when in China, gene editing was used to reduce the chances of a few children for getting HIV. And so there's been a lot of sort of banding of the field to make sure that we're actually using this in a responsible manner. The other application that I also want to highlight is the application of CRISPR for cancer therapy. So there's this amazing sort of immunological therapy called CAR T cancer therapy, which has been a way to treat certain blood cancers that didn't have a lot of treatment options. And how this works is T cells, which are a type of immune cell from patients, get taken from the blood and then edited outside of the patient and get edited to recognize specific cancer antigens that are specifically on cancer cells. Once that edit is made, then the cells go back to that same patient. So there's no risk of any sort of autoimmunity because that cell came from you and it just has a small change. And then those cells can go and directly target that cancer cell and not hurt any of the healthy cells. So that's another amazing example of how we can use CRISPR for things beyond disease states, but also for cancer. Another thing that I want to highlight in the space of infectious disease is that gene editing or CRISPR-Cas systems have also been used to produce what we call gene drives. And gene drives have been a technology that is used to actually limit the spread of infectious diseases that are carried by mosquitoes, like Zika or dengue virus or chikungunya, or even malaria. And the way this works is the change in the mosquito actually forces a way of driving the population to a certain type or genotype or certain type of mosquito. And that's demonstrated by this blue purple mosquito with the star. And how we've used gene drives is actually by forcing a particular species of mosquito into a state where it's infertile. So anytime that a mosquito mates, all of its daughter and son mosquitoes can no longer reproduce. And the reason why this is actually an okay application of CRISPR is because, for one, there are many, many mosquitoes in the ecosystem and many, many species, but actually only a small fraction of mosquitoes transmit disease. And because of that, just by making one population of mosquitoes infertile, you're not actually eliminating the rest of the mosquitoes in the ecosystem that do things like eat spiders. And the other thing that's cool about CRISPR is that scientists are actually trying to figure out how we can control these. So if we need to slow it down or back things up, we can. And so this is actually an amazing sort of newly developed application of CRISPR that could really, really help with our battling of infectious diseases that are spread by mosquitoes. The last thing that I mentioned, but is definitely far from reality, is that you may have heard that CRISPR has been proposed as a way to return the woolly mammoth to our generation. And that, I think, is a funny application, but we are very, very far from that. And so now I just want to back up and say, but what actually is CRISPR, and how did we find it, and what did Jennifer Doudna and Emmanuel Carpentier find, and how does this actually relate back to genomics? So all of these applications, whether it's gene editing or CAR T therapy or CRISPR gene drives, basically came from looking at the genomes of thousands of bacteria. And there are over 30,000 species of bacteria that colonize our planet. And there's also a lot on us. There are actually 10 times the amount of bacteria in our bodies, whether it's our gut or our mouth, our skin, compared to the number of human cells. And so there was a lot of bacteria to sequence and learn what exactly is in that DNA. And so what we actually found, what Jennifer Doudna and Emmanuel Carpentier and many other scientists found, is that within the genomes of bacteria, there's actually a record of past infections that are sort of stored into memory. And this may remind you of the way our immune systems work, where we have, antibodies that are a record of past infections that we have seen. And so this is actually, CRISPR-Cas was discovered as the adaptive immune system, or the memory immune system of bacteria. And it's one of the ways that it fights off pathogens. So bacteria are actually like us, they're infected by viruses. And in the field of bacteria, they're called bacteriophages. And those phages contain RNA or DNA, much like the viruses that infect us. And the way that these systems work is that the bacteria has that record in its genome of a a pathogen they've seen before. It makes that, and it surveys the bacteria for things that look the same. So it looks for a perfect pair, a perfect match between the record that was there and the new phage that's come in. And so when that perfect match happens, there's these proteins within the CRISPR-Cas system, which many of you may have heard of Cas9, which is the canonical CRISPR protein used for gene editing. And that protein, once it sees that this match has happened, can come in like a pair of scissors and cut up the genome of the bacteriophage and prevent it from spreading within the bacteria and hurting the other bacteria that are, close by. And this is the way that CRISPR-Cas systems are able to protect bacteria from infection. And so when I was a PhD student, I thought, well, human viruses, as I said, have the same sort of genetic material, whether it's RNA for a virus like coronavirus or DNA for like a herpes virus. And so I had this question of like, what if we used CRISPR in sort of its original form, which is to use it as a form of immunity or protecting us from pathogens? And so I asked sort of two questions during my PhD. I asked one, could we use CRISPR for treating infections? But also this sort of obscure thought of, well, what about for diagnosing them? It'd be really cool if we could, one, use CRISPR to figure out what virus is making you sick, and then be able to use CRISPR again as an antiviral. And so I'm going to start first with the antiviral side. And I think for many of you, this need for more antiviral treatments is quite obvious. We're more than 1 year into the COVID-19 pandemic, and our antiviral options are not enough and very limited. So we have currently one FDA-approved antiviral for COVID-19, and many of you probably know which one I'm talking about, which is remdesivir, which is the one that's been in the news and widely used. And although remdesivir is widely used and is the only approved, it's actually not, it's very far from a cure. It's only used for patients with severe disease that are in the hospital and just reduces the time of their stay in the hospital. So yes, it's a miracle drug for those people that end up in the hospital and helps them recover. And I'm very happy that we've been able to find this drug, but we need to be able to do better and faster. And so part of it, and this is the question I get a lot, which is why is it so hard to make antiviral drugs for COVID, but just all other diseases? Because actually for viruses, there's only a handful of them that actually have approved antiviral treatments, despite the number of viruses that we know we can potentially come and encounter. And there's many reasons, but I want to highlight just a few, which is that antiviral therapies need to disrupt the virus, so either inhibit it from getting into our cells, inhibit it from making copies of itself, but it needs to do all of that and not be very disruptive and have very many side effects. The other thing that we also see for things like antibiotics is that viruses evolve and they mutate, and therefore you need to understand when you give a certain drug, what is the propensity for or the risk of that virus developing drug resistance? Because then once that happens, you can't use that drug anymore. And then the other thing that's very challenging and relates to the fact that we're, learning about new viruses all the time is that new viruses, or not all viruses, actually behave the same. And that really makes it challenging. In contrast to the antibiotic field, we don't really have broad spectrum approaches or drugs for viruses because they just aren't all the same. And so this was a problem that I had hoped that CRISPR could solve. And so I started working on developing CRISPR-based antivirals for human viruses. And that work came out in 2019, where I and a team of scientists in the Salisbury lab showed that you could use a specific CRISPR protein, not Cas9, which many of you may be familiar with, but Cas13, which is a special protein that actually works on RNA viruses like COVID-19. We demonstrated that this could actually protect against flu. We did a proof of concept study. And similarly to what we saw with the mRNA vaccine, which is that as soon as that sequence was generated, scientists were able to very quickly show that a proof of concept that you could use Cas13, this CRISPR protein, to target SARS-CoV-2. And then in February, that work was pushed further and shown that it could actually work in animal models of infection. And why is this something that we could do so fast? So similar to the mRNA vaccine, the only requirement for development of a CRISPR-based antiviral is just knowing the sequence of the virus. As soon as you know the sequence, you can design that perfect pairing sequence that then Cas13 can come and cut up the virus and prevent it from future infection, much like what the bacteria are doing. But the one thing I do want to mention is that all of this work has been sort of a proof of concept. We've moved from working in the lab to trying this in animal models, but there's still a lot of work to be done, just like in the gene editing field, to make sure that this is safe and effective. And so there's a lot of work ahead to really make sure this is ready for use. So I expect that will take several years, just as it has for the Cas9 gene editing technology. So now what I want to talk to you about is this other sort of what I said was a more obscure application of CRISPR, which is the use of it for diagnostics. And this was something that, you know, CRISPR has been around for many years, but it's mostly been used for editing, and we've never really thought about it beyond the idea of treating some and making some correction. And so I want to step back a little bit and say that, you know, one of the things that I think about when I think about diagnostics is that anytime I'm sick, the first question that I ask myself is, do I actually need to go to the doctor? Maybe it's just a cold. I don't feel that bad. I'll get over it in a few days. And so most of the time, the answer to that question of whether or not I go to the doctor is actually no. And so most of the time, I don't actually know what is making me sick. When I say I have a cold, I don't really know exactly what virus that is that's causing a cold, or if it's some sort of bacterial infection. And so probably 90% of the time, my disease or whatever is causing my illness goes undiagnosed. But if I do feel like I need to go to the doctor, then there's sort of a second decision tree dividing line, which is that what is the way that we're actually going to diagnose that disease? Are we going to use symptoms, which is actually very effective in a lot of ways? There's a lot of times when you come in and the doctor knows exactly that you have a bacterial infection and gives you a Z-Pak and says, let me know if you feel better in a few days. And so in that way, your illness is diagnosed, but on a symptom level, so you don't actually know a ton of details about exactly what bacteria or exactly what virus. So if you don't go the way of symptoms, then your doctor gives you some sort of diagnostic test or says it's time to think about a diagnostic test. But for a lot of diseases, we don't actually have diagnostics for them. And so it's possible that even if you go to the doctor, that you still might not know what you actually have. And then, of course, in the other arm, for things like flu or now COVID-19, you can get a test and you can know exactly what you have. And so that's sort of the exact confirmation of what's making you sick. And so I want to bring up my next poll, which is, I think, was on my mind constantly during the early part of the pandemic, which is during the first wave of COVID-19 in the US, which was about sort of March, April, maybe a little bit before, depending on where you lived. If you sought to get a COVID test, how actually challenging was it to get one? And I gave you a lot of options here, as well as gave you the option to not say, if you don't feel comfortable answering. And as an infectious disease scientist and as someone who is working in diagnostics, I actually thought this was going to be something easy to get, because we knew for months that this outbreak was happening. And so I'm curious to hear how many of you either struggled or didn't struggle during these early stages to get a test.
I'll jump in here now and mention that the replies, 5% found it very challenging so far, 19% challenging but managed to get one, 19% it was simple, 57% didn't try to, and so far nobody said that they prefer not to say. So
that's good news. That's better than I expected.
So yeah, I'm surprised that 58% of the people didn't need to and didn't try to. But I guess I'm in that category.
I get it. If you didn't need to or didn't try to, I mean, we were all told to stay inside, right? And so if you really were strict on that in quarantine, then I could also see a reason to that you just didn't really try to, because you just stayed home and did what everyone was telling us to do, which was not go anywhere.
Yeah. And also our area, fortunately, did not have a lot of positive COVID cases, unlike, let's say, New York City.
That's very good.
In this rural part of the state, we weren't inundated with people being sick.
Great. Awesome. Thank you, Mary. So I'm showing you here sort of a shot from a drive-through test. So although it's great that a lot of you either didn't need to get a test or didn't find it that challenging, there were a lot of challenges, particularly in places like the epicenters of the outbreaks in the US, New York, Seattle, Los Angeles, where getting a COVID test was really challenging at those early stages. And in April 2020, we were actually only performing less than 200,000 tests across the whole US per day. And that's basically less than a 10% or 0.1% of the population. And because of that, because we just weren't testing as many people as we probably should have at that early stage, the numbers that we were seeing in case counts at that point in the year were probably a large underestimate of what was actually going on, and actually challenged our efforts to sort of halt transmission, because you don't actually know, who needs to stay home if you can't get a test in a timely manner. And the other thing that was actually really challenging for those of you that probably did get a test, and hopefully this was not the case, but 40% of individuals when polled that got a COVID test needed to wait more than 7 days to get their test results. And this is just like not good enough, because we all also heard that the sort of incubation period of COVID was around, 2 to 14 days, but for most people it was around 4 to 5. So by the time you got your test result, you either like weren't sure if you had COVID, or you already had symptoms and then you needed to stay home. And so that doesn't really help you change your behavior in the near term. And so there are many reasons why we struggled in the beginning, which I'm not going to go into, but what this should teach us for the future is that we really need to have a better way of getting tests and getting tested quickly. And one way that we really like to think about this is, what if we could just do testing at home? What if you could get a test kit mailed to your house that you could do at home, and in a short period of time get your result and know what you were supposed to do that day or that morning? And I kind of like to liken this to how we've gotten with pregnancy tests. You can pick one up at the drugstore, and within a few minutes you can do that test and know what the answer is. And so one of the things that I spent a lot of the beginning of the COVID-19 pandemic doing is figuring out ways to have CRISPR be a way of getting this at-home test. And so I've talked to you already about how you can use this special protein Cas13 that targets RNA to act as a sort of antiviral. But the other thing that we discovered when Cas13 was found is that once it sees this perfect pair and it becomes a pair of scissors and cuts it up, it actually turns into this like supercharged protein, which can then basically turn into a paper shredder and shred anything that's in its proximity. And it is this sort of paper shredding activity that only happens when that pairing is found that was exploited for CRISPR-based diagnostics. And we did that by introducing what I'm going to call a beacon, which basically like turns on only when that paper shredder is actually active. So if paper shredder is off and we've got this beacon up above it, it won't actually turn on. But then as soon as the paper shredder turns on, because it's found that pair, then a signal is produced and it says, hey, that virus is in the sample. And so this was very powerful, but how does this actually get us closer to the at-home type test model? And that's because one of the things that this technology does is that it actually does a lot of the things that a pregnancy test does. Your sample, whether that's a nasal sample or a saliva sample or a drop of blood, can actually be used directly in this test. You don't have to do any sort of fancy processing that happens in the lab, which we've heard lots about from PCR sort of tests. The other thing that's really amazing about it is you can actually turn this beacon into something that can actually be visualized by the naked eye. And so it actually kind of looks like a pregnancy strip where you have, a line one spot for if you're positive and a line in a different spot if you're negative. And the other thing that, is pretty amazing is that the results of these tests can come back in about an hour, and they're only getting faster and faster with the advancements of this technology. And so with these sort of three qualities combined, the CRISPR-based diagnostic field has really, gone full speed at trying to make a technology that can be used at home. And there are two actually startups that saw the promise of this technology way back when it was initially found in 2015, 2016. One called Mammoth Biosciences in San Francisco and another called Sherlock Biosciences in Boston that set out to develop CRISPR-based diagnostics for infectious diseases, for cancer, for human genetics. And they had been sort of working on early stages of these technologies, and then COVID-19 hit. And what's amazing about something like this, where we see a very urgent need for diagnostics, is that they sort of went into hyperspeed. And by the summer of last year, the first CRISPR-based diagnostic was approved for coronavirus in the United States through the Emergency Use Act. And this is, I think, just the start of what we're about to see in the diagnostic field about, how are we going to have technologies that can be used in the home so that we don't have to rely on sort of centralized testing facilities, and we can get results, not more than 7 days later. And I really am excited to see what these two companies will sort of push forward and really transform the diagnostic testing in the US and also beyond, because a lot of both of these companies have also expressed a dedication to bringing diagnostics to underserved nations as well. And so I just want to conclude with what I hope that I have convinced you tonight, which is that genomics and CRISPR have been very powerful technologies for understanding infectious disease, whether it's spread and changing how we're, preventing the spread of viruses like what happened in the Ebola epidemic, or whether it's, the use of these new CRISPR-based diagnostics and then maybe eventually these antivirals to treat new and emerging infectious diseases. But I also want to sort of look forward and say that, this technology relies just on sequence alone. And so it really has implications not just for infectious diseases, but also for human health as a whole. And so I think this is a very interesting area to, keep your eye on as we continue to discover new things and new ways to sort of use this for human health. And with that, I will again thank you all for tuning in, listening, and participating to in the polls, as well as thank you again to the Salisbury Forum for inviting me to speak tonight. It was quite an honor to be sort of in the names of all the amazing speakers that have come to the forum to speak to you all. And then I'm going to take questions, but I will also say I put my email down there in the bottom corner in case there's questions I don't get to, or you think of a question later on. You can always feel free to send me a note, and I will get back to you. So thank you again. And I'm
well, thank you, Catherine, so much for joining us. There are a couple of questions. I'm waiting for our audience to submit questions, but in the meantime, here, one just popped up here. Honey Sharp, why is it taking the FDA so long to approve certain vaccines?
Ooh, that's a great question. So there are a couple of reasons why it might take longer for the FDA to approve certain vaccines. I will say the one thing that's been really interesting about the timing of when all the mRNA vaccines went through their phase 2 and 3 trials is there were actually a ton of cases in the US of coronavirus. And so it really allowed them to evaluate the efficacy of the vaccines because there was enough COVID circulating that they could actually, people would get exposed and see how the vaccines versus the placebo were working. So that could be one reason why it's taking longer, because the trials are taking longer to evaluate. Another reason is just that depending on how the trials went, they may need to evaluate and have more testing in individuals to approve them. So those are sort of two layers of why it could take as long as it did. Lots of questions coming in.
Is funding, for current research in the field primarily coming from the public or private sector?
That's a great question. So in the US, a lot of the biomedical research is funded by the NIH and some of the sort of related government offices. However, at the same time, some of these high risk sort of technology driven projects can come from funding from the private sector. So I will say that, some of my work on antivirals came from Merck KGaA, which is Merck's company in Germany, but also the Sabeti Lab has gotten amazing sort of donations from philanthropists in the private sector to fund some of our sort of genomic sequencing work and other diagnostic work. So it kind of comes from both, but mostly public.
Okay, from Howard Fuhr, what does CRISPR stand for?
This is a good test of my knowledge. So it stands for clustered regular interspaced palindromic repeats.
Okay,
And the way so the way that name came up is because those records of past infection are in between repetitive palindromic sequences, and they're clustered together. So you get like one sequence that's repeated, and then you get that record, and then you get another repeated sequence, and then a record. And so they're clustered and they're regularly spaced, and the repeats themselves are kind of semi-palindromic. And that's where the name CRISPR came from.
Okay, from John Post, are there other applications versus cancer other than CAR-T? I'm not sure. RT. Okay.
Yeah, that's a great question, and unfortunately, a little bit out of my expertise. CAR-T is definitely the more popular one. A lot of the therapies a lot of the newer cancer therapies have been more in the immunotherapy space. So how can we use our immune systems to fight against cancer? And off the top of my head, I'm not thinking of a distinct CRISPR application, but I'm sure there is, and I will have to dig into my back of my brain. If it ever comes back to me, I will let you know.
From Molly Fitzmaurice, how might CRISPR be used to do early diagnosing of parasitic infections and their vectors as diseases such as Chagas move from Latin America north into the US?
This is a great question, and not something that I actually talked about. So one of the amazing things about this diagnostic technology, and one of the things that we showed in our early work, is that you could use CRISPR not just for diagnosing human infections, but also for figuring out what viruses are in mosquitoes. And we actually tested this on some mosquito samples that were collected in Florida during the Zika outbreak, and found that we could actually detect Zika from sort of mosquitoes that were collected there. And one of the things we think about a lot is not only should we use these diagnostics to sort of test someone who's sick and tell them what they have, but if we make a diagnostic that's, easy to use and cheap, then we can actually use it for what we call disease surveillance, which is what a lot of scientists are talking about, that we need more emphasis on disease surveillance in order to prevent the next pandemic. And that's just basically to understand what's circulating where we are, and what are we at risk of being exposed to, and Chagas is a good example of that. And so I think this technology is if we get it as cheap and easy to use, it will allow us to survey and see whether the vectors in our community are carrying these kind of infections. And this technology they're not limited to viruses. We can use it for bacteria, as well as parasites as well. So it's quite flexible.
Well, that sort of ties into the next question from Joseph Baxter. What exactly is the difference between a virus and bacteria?
This is a great question. So a bacteria is like a single cell organism in the environment, and it can actually live on its own. It can live on surfaces. It can pretty much live anywhere that's like amenable for its way of life. But a virus actually needs a host to replicate. And viruses are also much smaller than bacteria. So they're not what we call single cell organisms. They're in a lot of ways kind of like parasites. They need to be in a human or in an animal, or they need to be in an insect in order to live and reproduce. And so that's the main distinction between the two.
Okay, and we have gene editing seems like it could have a lot of safety concerns. What are the major ones, and what steps are scientists taking to address these?
Yeah, this is a great question. There's definitely a lot of talk about safety, and so there's safety on sort of two fronts. It's this question of, if we make a cut in our DNA, how do we know that cut's in the right place and not somewhere that we, don't want it to be? The other thing that we've been thinking a lot, and this is true of any drug field, is sort of like dosage. Like when we deliver this CRISPR protein into a human, how much is okay and how much is too much. And these are all sort of things that get tested along the research process. So there's been a lot of work to sort of figure out the best way to make these proteins very specific, so they only cut in that specific spot that you want. So you may have heard, when the Nobel Prize got announced, there's this talk about prime editing, which is, how do we make an exact edit? And this is, we've gotten as close to that as possible, or as close to that as we know of to date. And then the other thing, with safety is, again, this dosage, and that just comes from a lot of tests, sort of preclinical trial until now. And I think what I mentioned too is that a lot of these diseases that we're developing these therapies for that are in clinical trials just don't have any other options. And a lot of the patients are like, this is my best hope. And so I'm willing, to take the risk. And so we're so thankful for those individuals, just like the ones that, put out their arm for the mRNA vaccines back in early parts of 2020, is that, you do all that early research and then you have to make the jump. And that's why that jump is being made for things that just don't have any other options. And that will help us, further evaluate the safety.
I guess one of the things, this is sort of my own question, one of the things in terms of safety is you referred to the Chinese scientist who was sort of a rogue scientist. And because CRISPR is accessible and inexpensive, a lot of people, whether it's in different countries around the world, whatever, could potentially use this technology. And how do you deal with something that might be designed, whether it's a human or a plant or insect or whatever, that might get released into the world with unintended consequences? How do you police people from just doing kind of rogue research that could have a serious downfall?
Yeah, that's a great question. And I think, it's hard because in the case of the Chinese scientists, like he basically did it in secret. And so it's hard to stop something if it's happening in secret. But what I will say with that, and I think what comes to mind is the example of gene drives, which is that, the idea of gene drives is they sort of take over a population. But at the same time that we're doing this research on gene drives, we're also doing research on how to stop them and how to reverse the action of a gene drive. And so I think the combination of one having, sort of regulatory research bodies and regulatory health bodies that sort of, keep an eye on what scientists are doing as best as they can, but also making sure that, we have scientists that are doing the research from the other direction to say, what is something that we can learn how to, stop the bad actors if that does happen. And so I think sort of that two-pronged approach is the best that we can hope for right now. And I also think too is, the science community like really sort of bands together on topics like this. And so, I mean, you can't prevent all the bad actors, but the hope is that they're the very, very small minority.
Okay. Lloyd Baruti asked, could CRISPR technology be used to replace current RNA vaccines? And if so, could that preclude the apparent need to get boosters every year of the current RNA vaccines, which seems to be where we're headed?
That's a great question. Unfortunately, right now, the technologies that I described and the ones that I know that use Cas9 for viral treatment are not going to give us the same effect that a vaccine will for sort of long-term protection. But the hope is that, you know, we do seem to be going towards this direction of boosters, much like what we do for the flu vaccine. But I think, you know, if coronaviruses and COVID-19 are around for as long, we'll see the same push that we see right now with flu, which is that push towards sort of a universal long-term vaccine. So maybe in the short term, we'll need to get boosters now and then to protect us against the circulating variants. But eventually we'll get to a place where, you know, we have some vaccine strategy that will work against them all for a longer period of time.
Okay, Hartley Mead asks, can we CRISPR to vaccinate Lyme?
Like use the CRISPR to prevent the ticks from going against Lyme. That's a great question. And actually, I don't have a strong answer for this, but I know that, you know, there are former lab members of mine that are particularly interested in Lyme research, as well as other tick-borne infections, and, you know, trying to figure out new strategies, not only just to treat people with Lyme, but to, you know, counteract Lyme in the actual tick populations. And so maybe at some point we will.
All right, so Philip Oppenheimer asks, are there military implications?
This is another great question. So, so part of our work with diagnostics and also therapies was actually funded by DARPA, which is one of the Department of Defense technology arms, and they really were trying to accelerate the development of these technologies so that the military would have sort of like on the go diagnostic tests for diseases that they may encounter with on the ground, or be able to, you know, take a medication before going into a high risk area of certain diseases to protect our military. So there definitely are, and that is, you know, part of the reason why the Department of Defense was particularly interested in sort of these new approaches for treating infections and diagnosing infections.
Okay, so let's see. What do you see as the key changes that need to be made if we're to face another pandemic so that we can respond better next time?
That's a great question, and something I think about a lot. I would say, like, there are a lot of things that we could be doing better. On my side, the thing that you know, my former advisor Pardis and I talk a lot about is this idea of disease surveillance. Like, we really don't have a system in place to understand what is actually circulating in our communities, whether that's, you know, person to person, or whether that's the animals or the insects that are in the areas that we're living in. And if we had a network to test the environment, you know, cheaper and more easily, then that would help us basically understand what's there and what we're at risk of, what's at risk of, you know, turning into something problematic. The other thing that I think will be very important going forward is the use of sort of like community reporting of infections, where, like, we've actually seen this happen more and more with the pandemic, which is that, like, the Department of Health now publishes, you know, how much coronaviruses are circulating in our given communities. And if we think about using that as sort of like a community network, and you know, confidentially sharing, you know, oh, in this town, there's, you know, this many people that are either feeling these symptoms or, you know, have tested positive for this virus, then you can know, oh, you know, maybe I should, you know, refrain from this, or, you know, or maybe I should wear a mask outside to protect myself for a couple of days because the incidence of flu is really high in my area. And I think these sorts of community-based, you know, prevention of infection, as well as sort of more broad surveillance networks will, you know, help, at least on the scientific side of things. I can't speak for the policy.
All right, so Frank Fitzmaurice asks, what are the economic considerations when a disease originates in a poor country which does not interest big pharma?
Yeah, and that is a big issue. and we see that a lot for things like Zika and Ebola. Basically, as soon as the Zika epidemic ended, a lot of the big pharma companies cut off developing vaccines for Zika, which I put my head in my hands and was like, this is not going to be a problem that goes away. But unfortunately, they, have a duty to make a product that's going to be used for a wide population. And so what we need to rely on for the future for these diseases that are, impacting poorer countries is kind of a philanthropic network, or also having companies that have an arm that do sort of contribute to diseases that are hitting these poorer countries. And we're actually starting to see that with some of the companies. I'm more familiar with Sherlock Biosciences because it's kind of the arm that spun out of the diagnostics, but they have partnered with a foundation to bring diagnostics to poorer countries. And I think the more we see this, the more the better off we will be, because as we have seen, infectious diseases are a global problem. They're not, a one country problem.
Okay, so maybe this kind of leads into what you've been talking about, and perhaps this will be our last question. Can mobile and cloud technologies be a useful tool for helping stop outbreaks from spreading widely?
Yeah, definitely. That's a great question. So I would say yes, and it's basically what I just talked about. I think that if we have sort of an at-home diagnostic that you can, report on your phone, like I tested positive for this, and I'm feeling these symptoms, and that gets, reported to some sort of like almost like my neighbor community network, and we, learn more about what's circulating in our community through sort of mobile and cloud-based technologies, it'll just better, prepare us for stopping transmission and knowing sort of what we're at risk from. And so I hope to see, even though there's been a lot of pushback for sort of mobile technologies and privacy, they do have a place, and I think they have a good place in sort of preventing the spread of infectious disease. So I hope we see more of that in the future.
Well, I think just check in. I think that's the end of our questions. So I want to thank you so much, Catherine, for joining us tonight. It's been very enlightening, and thank you for all this the contributions you and your teams have been making to help keep us healthy. So anyway, thank you. I hope you have a wonderful weekend, and thank you everybody for joining us tonight. It's been a pleasure.
Thank you.
Bye now.